This video will begin our exploration of genetic linkage. Before we jump into that, though, let's have a quick review of segregation and independent assortment and get some basic terminology down. Recall that segregation is where two allele copies separate during meiosis, and only one is passed on to an offspring. In independent assortment, different genes with their own alleles segregate independently of one another. When this happens, you get both non-recombinant and recombinant genotypes and phenotypes in the offspring. Non-recombinants match the parents, and recombinants have a new combination of alleles and traits. So here we have a visualization for independent assortment. Like we've previously seen, two true breeding parents combine to produce dihybrid offspring. These dihybrid offspring can produce four possible gamete combinations. Let's zoom in on those for a second. When we look at those, we see examples of non-recombinants and recombinants. Non-recombinants are those gametes that match the parents. That is, the big R, big Y for round and yellow, and the little r, little y for wrinkled green. The recombinants, which are allele combinations different from the parents, are shown here in the red box and include yellow wrinkled peas and green round peas. Recombination here happens during meiosis, and specifically through the different metaphase arrangements of chromosomes during meiosis 1. These recombinations occur in the offspring in predictable ratios. However, not all pairs of genes show these same inheritance patterns and thus appear not to independently assort. Let's look at an example here of flower color and pollen shape. We start with two true breeding lines for purple and red flowers. The F1 is all purple with long pollen. Okay, so far everything looks like before, right? The difference arises in the F2 generation when we combine the dihybrids and look at the ratio. Recall that a dihybrid cross under independent assortment yields a 9:3 to 3:1 ratio. We don't see that here. If we look at the observed number, we see way more purple long pollen and red round pollen than we would expect relative to the purple round and red long. This is especially glaring in the red flowers with with round pollen, as this should be our least abundant group under independent assortment. But in fact, it's the second most abundant. So what explains this? The genes for flower color and pollen shape are physically linked on a chromosome. These two genes occur on the same chromosome and likely reasonably close to one another. Thus, they are inherited together instead of independently. So in our example here, we see way more of the non-recombinant phenotypes, which are those that match the parents here and match the parents here compared to the recombinants. But hang on, if these genes are linked, how do we get any recombinants? The answer to that is crossing over. This is the only way that linked genes can show recombinant genotypes and phenotypes. Recall that in prophase of meiosis 1, these genes are paired up next to one another, allowing for exchanges of DNA among homologous chromosomes, and therefore yielding some recombination of genes that occur on the same chromosome. Moreover, this recombination happens at a frequency relative to how far apart two genes are. And we will explore that in a later topic.